Hello, everyone. My name is Brandon Dimick. I am a PhD student over at Duquesne University, and today I want to talk about some of my PhD work relating to prostatic acid phosphatase in hominids. Oh, there we go. Um, so the main focus of our lab is studying hominid mating systems and how that relates to varying degrees of sperm competition and how that may be a selective pressure when it comes to seminal proteins. So what we could do is group species based off of sperm competition or degree of sperm competition. The three that I'm going to be focusing on for this talk is human, gorilla, and chimp. So humans and gorillas, we are both low sperm competition species. So humans can be either monogamous or polygynous, depends on the culture. Polygyny is also found in gorillas, where you have a single dominant silverback male mating with multiple females. So in any case, you have relatively few males vying for each female when it comes to reproduction. And then on the other end of the spectrum, we have a high sperm competition species with chimpanzees. They are polygynandrous. You have multiple males mating with multiple females. And as you can imagine, that is a very strong select pressure for when it comes to sperm competition. So with polygynandry and this high sperm competition, you see a number of morphological and physiological changes, including the formation of a copulatory sperm plug, which is where when a male ejaculates and actually the semen coagulates, it actually gets so rigid it actually prevents other males from being able to fertilize the egg. So that is one change that is a result of high sperm competition with chimpanzees as well as bonobos. But focus of this talk, I'll stick with chimpanzees. So what about at the protein level or the gene level? So the protein that I am researching is prostatic acid phosphatase. It goes by a number of different names in the literature. For shorthand, I'll be referring to it as ACP3. We know that it functions as a homodimer. The dimer is about 100 kilodaltons in size, with each subunit being about 50 kilodaltons. We see high expression of this protein in prostate tissue, and it is abundant in seminal fluid. It has a number of targets, including semagellins, which are involved in semen coagulation and liquefaction, and we know that it targets phosphocholine, which may be involved in sperm motility. However, if we can pinpoint one canonical function, that remains to be seen. Perhaps it just has a number of different functions. So when it comes to seeing if this protein is being impacted by sperm competition, what we want to first answer is whether or not there is even selection occurring at this gene. So to do that, we looked at DNDS ratio. So I calculated these values using a free ratio model in PAML, and then I plotted these values onto a phylogenetic tree of primates. And what you'll notice circled in red here at the top is that here we have human chimp gorilla lineage. In the human lineage, going back to the human chimp gorilla common ancestor, there have been no non-synonymous changes. So that strong indicates very strong purifying selection, hence the 0 value. Gorillas likewise also have purifying selection with that 0.22. And then chimpanzees, we are seeing evidence for strong positive selection of ACP3 with that value of 2.19. So there's more non-synonymous changes than synonymous changes. So how does this impact the protein? Well, we're finding that it impacts the protein in a number of ways, including in expression. So here you see these bands from a Western blot from two different individuals when it comes to each species. So 2 individuals from each species, and then the relative abundance of ACP3 in their seminal fluid was calculated using both Western blot methods as well as LC mass spec. And what you'll notice right off the bat is that we're seeing a slight decrease in expression when it comes to chimpanzees, so 2 separate individuals. And then gorillas, we see a drastically diminished relative abundance. Actually, you can only detect it, at least in the Western, in 1 individual. So what about the protein itself? So there are 4 amino acid substitutions, non-synonymous amino acid substitutions on chimpanzees. And One in particular I'm very fascinated in. It's this I-130. So before I go forward, what you're seeing here is a human model for its X-ray crystallography structure, and I just mapped the changes on the human model. And remember, it's a homodimer, hence why you're seeing each two arrows coming from each substitutions. But what you'll notice is that I-135 to V-135, that is 3 amino acids away from a critical homodimerization residue. And we actually see that this site in mammals is often a target when it comes to positive selection. So there's that in chimpanzees, which is going in line with the high DNDS ratio. And we also see some other sites, such as that I-277 to T277 in that linker region. To the right and to the left, we have that T233, the I-233 in that alpha helix. And then there's a beta pleated sheet where we see an L315 to F315. Gorillas, there's actually three amino acid substitutions. And at the top, you see the C terminus. That's where the third is. I didn't map it on this X-ray crystallography structure because I couldn't map it. It's on that C terminus at the top. But what we can see is that K185 to Q185 and that alpha helix. And towards the bottom of the structure, there's that R86 to K86. Those are also in alpha helices. So yes, we do see these substitutions, and I'm able to map them. In the future, I'd actually like to do some site-directed immunogenesis to see which of those changes may impact function more than that in a little bit. But well, first question, how do we actually get the protein? So what I do is some recombinant technology. So human chimp gorilla gene is inserted into a plasmid, a human expression vector. I then transfected these plasmids into HEK293 T cells. And then 72 hours later, I collect the protein that is hopefully within that cell media. And we did have successful expression of our recombinant proteins based off this anti-ACP3 Western blot. Positive control lines up, and then all of our samples do show up on that Western blot. Now, to actually quantify, we used an anti-ACP3 human version for ELISA. We used an ELISA, and then we normalized using a Western blot method. So what I did was I ran the human chimp gorilla samples in triplicate, quantified these ACP3 bands, then averaged them, and then with also a loading control, we then normalized those values down to the lowest band intensity. And then I run it again using those normalization values essentially as dilution factors. And then using those dilution factors, I normalized. And lo and behold, this is after the second Western, we do not see significant differences between our samples, which is great. Now, when it comes to actual assays, I did two varieties. I did a non-native substrate and a native substrate. So when it comes to the non-native substrate, I used a very popular substrate Dimick when it comes to phosphatase assays. So that phosphate group is cleaved and you get a fluorescent product. The first thing I wanted to answer is, do these three species share a pH optimum or when it comes to the three orthologs? And what I found was all three orthologs share a pH optimum at pH 6. And we see varying activity from pH 4 to pH 7, no activity beyond that, which makes sense physiologically. And what's interesting, though, is you're already seeing, we're hopefully seeing that there's this statistically significant difference between these three species. So I really wanted to look deeper into that phenomenon. So to do that, I did some old school Michaelis-Menten kinetics, varied the diff up concentration while also calculating initial velocity to calculate Vmax, so maximum velocity, Km, or Michaelis constant, how efficient is the enzyme binding to its substrate, so the lower the better. And then Kcat, and then Kcat over Km, which is measures of catalytic efficiency. And what we notice is that chimp is showing to be the most efficient enzyme across the board. Then human is somewhere in the middle, and then gorilla we're showing a relatively low activity. And then when we look at Kcat over Km for all three species, we do see statistically significant differences there, which is interesting. Then we also want to test it on a native substrate. So in this case, we used phosphocholine. It's been used in the past as a great substrate for prostatic acid phosphatase. We do see rapid dephosphorylation of phosphocholine post-ejaculation, most likely because of ACP3. And there's actually a large quantity of inorganic phosphate as well as choline in ejaculate. And the reason for this is because the choline may be involved in reactions in the mitochondria. They have an enzyme called choline dehydrogenase. And as you know, or probably know, sperm cells have a large number of mitochondria. So we thought this was a great target for our assays. So to do this, I measured the release of inorganic phosphate using a malachite green assay. So when inorganic phosphate comes off of the substrate, it binds to a dye and it makes a nice, pretty green color. And these are some of the results from those assays. And we're seeing similar trends to the DIFMUP assays, where chimp is showing the highest maximum velocity, humans are somewhere in the middle, and then gorillas show the lowest maximum velocity. And keep in mind the large error bars. That's just because this was run in duplicate. Of course, before publication, I'm going to increase the number of replicates to hopefully tone down on those error bars. But we are seeing this consistent trend that is also lining up with our DIFMUP assay. Now, remember, those changes are derived, they're lineage specific. So those 3 or 4 changes in chimp are lineage specific to chimp, and then gorilla, there's those 3, they are specific to gorilla. We know this because there have been no non-synonymous changes since the human chimp gorilla common ancestor. And the results that we're seeing are lining really well with what we expect based off degrees of sperm competition. Chimp being a high sperm competition species, we're seeing an increase in activity. Humans are somewhere in the middle, and then gorillas, they're very low sperm competition species. We're seeing this decrease in activity, and we see this in other proteins as well. For example, KLK3, which is another seminal protein, we're seeing increases in activity or decreases in activity. And then another example, Magellan-1 is actually becoming a pseudogene. It has a premature stop codon. So this is actually lining up with pseudogenization due to relaxed constraint. So some key takeaways that I want you to come home with are chimpanzee ACP3 has evolved to be relatively more efficient than other orthologs. And this is regardless of the fact that we're seeing decreased protein expression, we're still seeing this increased efficiency. So perhaps chimps have evolved to do more with less protein. So they're using less resources to make protein. Gorilla ACP3 is functioning with decreased phosphatase activity. So we're seeing a decrease in protein expression. So perhaps it might become a pseudogene like some other seminal proteins. These changes are following patterns that appear to be due to variation in degree of sperm competition. So these patterns are starting to line up here. And the aforementioned evolutionary changes are lineage specific. So some of the questions I want to continue to answer is, for one, really get home is it actually sperm competition? The patterns are there, yes, but I'd like to actually prove it, or at least support it or reject it, see what happens there. And I also want to know which of those amino acid substitution caused the changes in activity that we saw, as well as if there are differences in abundance of homodimers. Because remember, I mentioned that one amino acid residue that's near that homodimerization residue in chimpanzees. I'd like to know if the increase in activity is at least partly due to an increase in the abundance of homodimers, which that would be very interesting if that was the case. So just a couple acknowledgments. I'd like to thank my committee, my boss, Mike Jensen Seaman, as well as our undergrads and grad students. And I'd also like to thank my colleagues, Emine and Bree, for their help with the panel. With that, I'd like to thank you for stopping by to listen to my talk. I'd be happy to answer any questions. So if you go under my presenters, you can find my email. You can send me any questions. And I hope you have a great day.